also the ITN CONTRA PhD student, Monica Valecha, that she's doing a PhD at David Posada's group in Vigo.
Thank you, Hania. So today I'm going to talk about my project, which started as a side project, but somehow turned out to be one of my main projects. So I'm going to talk about benchmarking the single-cell DNA somatic variant callers. So we have heard with many talks, or most of the talks, how important mutations are. And especially given the single-cell data, you need mutations for all kind of analysis, for all downstream analysis. Maybe it is mutational signatures, phylogenetic analysis, or while Nico talked about the molecular clock, selection, therapy, and many more. So I think it's fair to say that mutations are a starting point for many of you sitting here, but it is very important to have those reliable mutations. If you see single-cell sequencing data, it is full of noise. In the above plot, you can see that the bulk sequencing has very uniform coverage, and the variant calling is very standardized. But that's not the case with single-cell data, because you have a lot of errors, as showed here. Like you have uneven coverage, you have extreme cases like allele dropout, you have allelic imbalance. You can also have a locus dropout where there is no information, or you can have amplification errors, which are introduced by different whole genome amplification strategies. So we thought it is important to benchmark the variant calling here, because we all need mutations. And when I was analyzing the data, because our lab concentrates on a lot of real single-cell whole genome data sets, I came across a lot of mutations which just did not seem right. Maybe with the frequency, they seem like errors, or just with even if by merely looking at numbers, we saw that this cannot be true. So we went back to seeing like how can we reliably identify mutations from real data sets. So to perform a benchmark study, we all know we need some simulation data. So this is an independent study, because we are not developing a caller here, but we will use other people's developed caller in our study. So we wanted these simulations to be unbiased to any caller, but also to be very close to the real data sets. Because the thing is, callers do have simulations, but they fall into this self-assessment trap that they simulate their data based on the assumptions they are making in the caller. So even if you can say it's a good thing, but it gives a unsaid advantage to the caller they develop. So we wanted to keep this study as independent as possible. So first we simulated the sequences. We introduced mutation based on the cell genealogy. We got the sequence from 1,000 genomes. And then we took real single-cell data. So in our lab, we have a lot of single-cell data. So what we did is we took the coverage profile and allelic imbalance profiles from this data, and we introduced it into our simulated sequences. And so basically, these simulations are learning everything from the real data. And in the end, we simulate the sequences, not the read counts and not the mutations, because there are many simulators which do the read counts, which will tell you the truth. But to use the variant callers, you need reads. You need the BAM files, alignment files to start with. Then our model learns coverage profile and allelic imbalance from the real data. Here you can see this is a cartoon showing Ampli1, and I will show you further that we have also used different kits for the same. And then we also add amplification errors, and then we merge the reads, and we have this simulation design. So in this design, we have 10 tumors, or in technical terms, these can be our 10 replicates with 20 cells each. This is whole genome, chromosome 1, whole genome strategy of chromosome 1, because chromosome 1, in our opinion, was more reliable to represent the whole genome data set, because many people consider chromosome X or chromosome 21, but then it is very hard to scale that data. Then we had 16 different scenarios for whole genome amplification kits. We used two major kits. One is Ampli1 and another one is Repli-G. You may have heard from David's talk yesterday that these two kits have very different biases and very different recurrent errors. So Ampli1 is good in a way if you want to do a phylogenetic study because it has all the recurrent errors, though the coverage breadth is less than Repligi, but Repligi does not amplify across the same regions. Then we have two allelic imbalance levels. By these two allelic imbalance levels, we mean that the real data has allelic imbalance, but also there might be cases where we don't see that allelic imbalance that profound. So we wanted to see how the callers perform when there is no allelic imbalance. Then we did this at 4 different coverage levels, 20x, 10x, 5x, and 1x. You might think, why 1x? Because it is very less to call mutations. But if you see the data out there, like from Nick Naveen's lab and other labs, you have 1x data because they are concentrating on CNV, which is good. But you cannot use the same data for SNV. And doing at 20x is a very costly affair. I mean, PIs who work in single cells would know that. But let's say it's not practical or feasible to have 20x cells because we are talking about whole genome or whole exome, and maybe even 20 cells are sometimes less. Maybe they be...
For whole genome, it's impossible. For whole genome, it's less than 1, actually. 1 is the law.
Yes, for the data sets out there, but if you want to do a mutational analysis, you cannot use 1x data.
Yeah, but then you don't have the whole genome. You just have to target the whole exome.
Yes, but then you lose a lot of information. Plus, given the single cell noise, you already have a lot of missing data and other things. So it also depends on your study. So someone who wants to do mutational signatures, whole genome would be a better idea, right?
Would be great, but I don't think it will work done.
So,
We wanted to, this is a great study abstractly, but essentially we know that the current data can either be whole genome lower than 1x and you never use it and everybody knows it. You don't use it for mutation coding. You only use it for copy number coding, or it is less than not whole genome and it is higher coverage, and then it's actually, this data is also differently generated because the first one has even coverage, so it doesn't have these, it really doesn't have, I think this was also what David somehow was commenting, but it really does not have this data which is less than 1x, no, it does not have all these amplification problems. But then the other one, which is the whole exome, has higher coverage, and then it has a lot of amplification.
yes. And in our lab, we have whole genome sequencing data for colorectal cancer for, say, 6 to 10 patients with 20 to 100 cells, which are whole genome, and we are also doing at 5x.
Whole genome 5x.
Yes. I mean, the one we shared with even Steve and so.
You have these
Yes. Variation, yes, that's true. But there are even public data sets out there that, have whole exome data sets.
Whole exome, yeah.
Yeah, and we also have neurons data set, which is whole genome, and one pancreatic cancer data set. So, of course, not all the people or many people are doing it, but people are going towards it for phylogenetic analysis and mutational signatures. That was the idea behind simulating the data at different x to see how variant callers perform, because we are already performing this study. So it is easy for us to downsample and see what coverage effect, puts in variant caller.
Yeah, no, I think it's very interesting analysis. I wonder if one of the main problems is amplification errors.
Yes.
You simulate amplification.
Yes, so that also we learn from single cell data, but here we are using normal cells. First, we are trying to identify mutations in these normal cells. Then we use a beta binomial model to simulate those errors because we were getting a lot of flat signatures here. So we take those and then we try to put them in the simulated sequences. And with this, we also keep in mind that we try to remove as much as possible the true mutations based on the VAF.
Yeah, the copy number, you don't include the copy number.
So copy number also we include, but if you, I will show here. So we are actually starting with sequences as our starting point. So for copy number segments, we just introduce as them as the segments, and then we merge the whole system.
So you like simulate some copy number event?
Yes.
Okay, because you haven't mentioned.
No, I did not mention it because we are concentrating on SNV, but we do simulate copy number, and the simulations in itself are very complex. So, I mean, I forgot to even mention my colleagues that help me with the simulations here, but I think I will run out of time in that scenario. And yeah, but I'm happy to talk about the simulations, and we can discuss more about it. So in all, we had like 3,200 single cells, which includes 10 replicates, 20 cells each at 2 different kits, 2 different AI, non-AI aware, and at 4 coverage levels. Coming to variant callers, we know like there are 7 published variant callers and few are in pipeline. So we just, when we started this study, we just went with the published variant callers right now. These variant callers have different strategy they use for calling the variants. Like you can see in that first block, Cornbase and Lyra use read phasing strategy. Then we have scan SNV and SC caller that use allelic imbalance modeling. With the HSNP, here the HSNP is the heterozygous SNPs present in the data in order to call the real variants. Then you have sci-fi and other which are still to publish or in bioarchive that use the phylogenetic inference in order to call variants. You can read them, all of them are published. Let's go to how these variant callers were actually performing. These are very preliminary results. You can see we have Ampli1 and Repli-G there. The upper blocks are the precision, and then you have the recall here. You can see some callers have really good precision, like Lyra, Scan SNV, and SC caller. But then the overall recall of these callers we are talking about is like less than 50% or 40% in that case. And that is understood because you lose, Yes, that is because you do lose a lot of data, and plus the breadth of the data is not 100% in single cells. So that's where you kind of don't get a very higher recall here because your genome covered is only 50% or 70%. And some tools like Scan SNV, it does perform very good. It only gives you all the true positives from the data, but then the recall is less than 10%. You have SC caller where you have a greater recall and it performs precisely well. So basically, it also depends on what question are you asking, you know. You want very few true mutations, or you are kind of considering to give a precision recall balance somewhere, no? And you can see like this is very important, like SC caller is performing better in Ampli1 and not in Repli-G. But then that is coming from the kit. You remember I said you like Ampli1 has recurrent errors, and phylogenetic aware tools need that information. So maybe Ampli1 using sci-fi or even for maybe for Sieve in future would be important because they have the same regions, which is not the case in Repli-G. We do see some behavior that I cannot explain right now, but I will start conversation with the authors in order to explain that behavior. Because SC call sci-fi, it has a very good at 1x and not very good at 20x. But that can be explained, maybe the number of false positives there is very high when you go with a higher coverage. And yeah, so this was at different sequencing depths. And Cornbase,
X, and none of the methods has recall higher than.
That is the tragedy. And this is not something just in simulated data. Eventually I'm going to add a lot of real data to it, and that is what this motivated this whole study, right? That either we are getting a lot of calls from monovar, which are mostly false positive, or we are losing a lot of information if we use precise callers like SC caller and Scan SNV, which were like two mutations, what do you do? Yeah, I'll hurry up. And then there is this allelic imbalance performance. Most of the single cells show allelic imbalance, and we also have a model where there is no allelic imbalance. Callers slightly perform better, but I would say it's more or less similar performance in both these cases. This is one of the applications, and I only included this application because most of you know I started with mutational signatures from single cell data, and it is still a very important part of my PhD thesis. So we tried to see what are the signatures lying in the errors. So we enumerated these false positive errors, and we see that a lot of callers in the number of thousands are giving, flat signature like SBS5. So we have more downstream analysis to follow, like how they perform on phylogenetic inference and so on. But I'm not going to show that. Please stay tuned. We will publish the results hopefully soon. And maybe because I'm running out of time, it is just worth mentioning that these scholars take a lot of CPU time. But of course, given your computational architecture, you can always kind of play with it. So in conclusion, the aim behind this study was first for the lab itself as well, to identify reliable mutations from so much single cell data that we have in hand, but also that it will help people to choose a caller for reliable mutations, like make an informed decision basically. Then what is your use if you want to do somatic calling or germline calling? We tried to keep our simulations realistic. Not many people simulate actual sequences. Then we also have false positive classification based on callers. And there is no other study which is showing a benchmark of single cell data. And then what is the ADO effect on callers? Because ADO is a very important part of single cell sequencing. And I would like to thank my lab members and also my collaborators, which through different projects realize the problems that we face and are there to help. Thank you.